So I arrived at the LMB, Laboratory of Molecular Biology, shortly after Easter in 1974, and quickly became acquainted with and in contact with an extraordinary group of people. Francis Crick, Sidney Brenner, Hugh Huxley, Fred, of course, and Max Barutz, and Aaron Klug. But the ones who shone publicly were Francis and Sidney. They dominated all discussions at seminars. They dominated the questions, and indeed Francis would rephrase questions that people put, because he thought that he understood the question, what the person was trying to get at, better than the question themselves. And actually he once did that to me. I even remember the question that I was asking, because it impressed me so much that anybody would do this.
Did he rephrase it better than you'd?
Well, no, I don't think so. I thought it was particularly clear and succinct. Still do. Actually related to the number of ATP molecules that were consumed in packaging DNA into a viral head. It turns out I end up working on the homologue of the molecular motor that does this. So it's something that's come back into my life in the last 15 years. But I was asking this question in 1974 or 5 when Francis was still there. He left, of course, soon after, and went off to the Salk Institute and severed his links, formal links to the Laboratory of Molecular Biology.
And as a thinker, I was very impressed in my interview of Aaron as a very brilliant mind.
He did. He is a brilliant thinker. He has been a brilliant thinker, and he has a very wide knowledge. I learnt quite a lot from Aaron in the 1990s. He got interested in Alzheimer's disease, and put together a group of people at the LMB to look at bodies that are found associated with people with Alzheimer's, who died from Alzheimer's disease, called tangles. These are tangles of proteins. It wasn't known what they were made of, and it seemed that there was an association with the presence of tangles in Alzheimer's brains and their formation, and so in order to try and understand the origins of the disease, understanding the origins of the tangles seemed to be important, so you had to know what they were. And so he put together a team which involved César Milstein, myself, and then a medical person called Claude Vichik, who had access to the material. I was doing the chemical analysis, César was doing immunochemical analysis, and Michel Godet was also part of it. And Aaron managed this group of people, and he had a particular managerial style, actually, which I'd never encountered before, that I found really quite interesting. The way that he took people who were really quite senior. I mean, César was already a Nobel laureate, but he managed that group of people on this particular topic, and it was quite effective, and we also did this in our spare time, as it were. But Aaron kept us together, and we achieved something together that was quite important. It was published in 3 papers in the PNAS. So I learned something about managing groups of scientists from that experience.
What about César Milstein, who you've mentioned?
Well, I knew César very well because he was in the next laboratory to me. So he was in the division of protein nucleic acid chemistry, and he wasn't Fred's PhD student, as some people have written. That's incorrect, actually. He worked for a man called Malcolm Dixon. But it was Fred who encouraged him to come back. He left Cambridge and went back to Argentina after his PhD, and it's Fred who encouraged him to come back and to work on immunological topics, which is what he did in the Department of Biochemistry, and then moved down to, I think he'd gone back to Argentina again, but then he came to the LMB and worked in Fred's division. Cesar, a delightful person who had a really quite illogical mind, seemingly illogical mind, and this infuriated Sidney. Sidney was a very logical thinker, of course very rapid, but it was all based on facts, and well, of course Sidney made jumps, but Cesar somehow would make wild jumps that often turn out to be correct, and he wasn't very good at explaining where he was coming from or where he was going, but experimentally he was just somehow extremely good and extremely effective. He was very knowledgeable. He knew all about his own field. I liked him a lot as a person and a scientist, but again I came into conflict with him through him being Fred's successor, head of division, because Cesar wanted to focus the whole division on immunology, and yet that's not what I did, and I managed to persuade Cesar not to change the name of the division to Division of Immunology, and that it should remain as it does to this day, protein nucleic acid chemistry, so that it's a broad church and can incorporate all kinds of different people, and I think that was the right thing to do. He wasn't very supportive of me through difficult times, whereas Max in particular was. Fred had gone. I mean, when I went to Fred to tell him what I wanted to do, he just listened very patiently and said, "Well, why don't you get on with it?" Which was, that was the equivalent to writing a grant for several people for 20 years. But he did. I mean, he did support me, and once it became clear that I was going to be successful, Cesar became extremely interested in me, actually, and was very happy about my success, although I still feel that he didn't really help me by helping to engender that success in a very positive kind of way. I don't wish at all to be unkind to Cesar, because he was such a pleasant and nice person.
Just, I'd like to come to your success in a moment, but just two last people who I've interviewed, two other chemists, Kingsman, who you mentioned. One was Hal Dixon.
Yeah.
Did you know Hal well? You said you met him in 1974.
I wouldn't say that I knew Hal well. I met him during December course in 1974. This was the first time I met him, and was interested in his very deep knowledge, actually, of chemistry of proteins. I think I benefited from that. But my contacts with Hal really were very few, and over the years we never did anything together. I just meet him very occasionally.
What about Dan Brown?
Dan different. I mean, Dan retired, took early retirement from the chemistry department, and then came to the laboratory of molecular biology as a retired worker. And so I used to talk to Dan quite often. We once nearly did something together, but we didn't quite. It required the synthesis of an unusual nucleotide. But I think what happened was I managed to solve the problem by a different route. We did some experiments together, but I liked talking to Dan, and I found he had an interesting view on the world, and he, I think I mentioned earlier, he had insights into how the structure of DNA was worked out by Francis Crick and Jim Watson. He also would say quite interesting things about Lord Todd.
Really?
Well, he'd worked with Alexander Todd, and he could be quite critical, I think, at times.
I missed that on his interview, but he did talk a lot about RNA in his interview, and his work on that seemed to be rather important?
That I'm not so familiar with. Dan's contributions in that area. That was long before I came. I mean, I'm aware of work on RNA that went on in the Maltino Institute from Roy Markham and John Smith, with whom Dan interacted with, both of them. I know in general terms what they did. It was to work out methods of separating oligoribonucleotides by chromatography, and I think probably Dan's contribution was how to break down the RNA in a controlled way, but I'm not certain about that.
One last point. When I interviewed Aaron Klug, he was very keen to talk about zinc fingers. Seemed to think a lot was potentially there in the future with zinc fingers. You must have come across these.
Well, I remember their discovery by a student of Aaron's. Didn't discover them, but he isolated the material from frogs, from Xenopus frogs. There was this huge slaughter everywhere. This guy went from which he got material from which he isolated the protein in which the zinc finger was identified. I think it was Aaron certainly put the picture together of what was going on, and its importance as an element in many transcription factors. Aaron used to claim that the zinc finger was the most abundant motif in the protein world, but I'm afraid he's wrong, because the most abundant motif is called the Walker motif.
Is it?
That I discovered. I once said this to Aaron, and he said, "Are you sure you're right about this?" But I've actually been collaborating with some bioinformaticists who confirmed that it is indeed true. So this is a motif that's associated with binding nucleotides, with purine nucleotides, ATP and so forth.
Well, tell us, that's an obvious lead in to the end of the story about how you got your Nobel Prize.
Yeah, so I started to talk about Peter Mitchell and how the bioenergetics field was dominated by him and his concepts of chemiosmosis, which have been very difficult to get established, and even after his 1978 Nobel Prize, there are many people who still thought that it wasn't correct, that the proton motive force, as Mitchell called it, was not. Well, Mitchell's view, which is correct, is that it's an intermediate in energy conversion, but there were people who disputed that, and so through the period that I entered this field, it was still dominated by debates on chemiosmosis, and particularly on how proton pumps work, but without any discussion or experimentation to really understand the fundamentals of the pumps themselves. That came later. And Mitchell sort of was very insistent on experiments being done when what he did was he hung on to ideas too long, and this forced other people to keep on doing experiments that they might not have done, and which probably were not very profitable. So his general concepts were correct, but some of the details that he'd said over the years turned out not to be precise, and that was true of the ATPAs that I worked on. So I got on with the ATPAs from the early 1980s, and there have been some very good biochemistry done, but none of the proteins have been characterized properly, and so I set about sequencing them using the old-fashioned, by current standards, protein chemical methods. That I quickly latched on to using recombinant DNA. Actually, Sydney helped me there and provided me with material that was extremely helpful. It's one of the good interactions that I had with Sydney. Fred didn't want me to use DNA. He thought, I've employed you as a protein chemist, and you shall study proteins and not nucleic acids, and he wanted me to develop methods with proteins, and I mean, I disagreed with him, but he let me get on. I said, well, I was going to use them anyway. He said, you probably get beaten. Knowing perfectly well that this would spur me on to even greater efforts. So Fred was pretty good at psychology, I think.
Yeah.
And so I worked on establishing the chemical composition of the enzyme, number of proteins in it, and discovered many components that were unknown, reconstituted parts of it. But my aim was always to get a high resolution structure, and so from the very beginning I was trying to grow crystals of, at the time, what people thought was a very large protein complex, and many people thought it was impossible. And I was variously advised that I was wrecking my career, and why didn't I do something easier, and it just preposterously difficult, and so on and so forth. But this is exactly, I think, what appealed to people like Max, was that I would be prepared to take on something like that, and seemed to be making progress, and so they backed me. And
were there moments when you despaired? Or?
Never, no, never. Because we were incrementally making progress the whole time, and I was in a community of people actually who didn't think this was unusual to struggle with a problem for a long time, as long as it was an important problem and people could see that it was, and I seemed to be doing things apparently in an appropriate way.
When you say a long time, how long a time?
Well, I mean, I think I started trying to grow crystals in 1982 or 3, and by the time they were any good was 10 years later. And I kept at it the whole time with a particular postdoc, René Lutter, from
how do you spell that?
Lutter, and with a research assistant, Michael Brunswick, we kept going the whole time, and things got. We had crystals from the very beginning, but they didn't diffract very well, and I didn't know much about growing crystals, let alone how to diffract them, and eventually we sort of learnt these skills. So I've always said that it's very important to have an important biological topic, and then problem, and then use whatever techniques you need to solve it, and not to be frightened to learn new methodologies. And so, I mean, it all worked out in 1994 when we solved this high resolution structure. Then it became, when I looked at it, it became clear that it was very important, because one could interpret the catalytic cycle in a way that we could not really have hoped to have been able to do. So we could relate the structural information, so these were single snapshots really taken by X-ray crystallography, but we could work out from them a great deal of information that linked to work that had been done by Paul Boyer, which related to the way in which ATP was being made. And from this it was inescapable that the enzyme was driven by a rotary mechanical mechanism, which was something really quite new. So I've not explained the background. So the issue with Mitchell was, how was the proton motive force linked to the generation of ATP, a chemical synthesis? And Mitchell's view was that the protons went across the membrane, the inner membrane of the mitochondrion, and they participated directly in the catalytic site of the enzyme in phosphorus chemistry to produce ATP. So this he called direct or chemical coupling, and another chemist at Oxford called R.J.P. Williams had similar views. And the catalytic site is a long way from the membrane, and so you have to transport protons through a proton wire, which would be 150 angstroms, through an aqueous space, which seemed pretty unlikely to me. And so the alternative view was called indirect coupling, but Mitchell was dead against indirect coupling, right up to his death. He was much more, he was strongly in favour of this idea of direct coupling, as he called it. Anyway, it turned out that it was indirectly mechanically coupled, and that was a new concept in enzymology of a rotary action produced by consumption of what is really possible, which is electrical energy, the proton motive force being used to generate a rotation, rather like water flowing through a dynamo generates electricity. It's a rather similar kind of action, actually. And so that, I mean, this made a big splash, and it now turns out, what is it now, 15, not quite 15 years on, that this rotary principle is really quite widely dispersed in a way that hadn't been realised, and the machine that I was trying to ask a question about in 1974, that Francis interrupted me, actually belongs to the same family of rotary enzymes, and so I think that's been pretty fulfilling to see that happen. The motif comes back to when we were determining a lot of protein sequences, and we knew that some of the proteins bound ATP, and we wondered whether we could work out in this 500 amino acids which bits were involved, and so we started comparing sequences that we determined with enzymes that were known to bind ATP, and we found some similarities. At that stage, the mathematical methods for doing this kind of calculation were not very well developed. The best method was devised by, again, someone at the LMB called Andrew McLaughlin, and we use Andrew's method to do this. Today people would not find what we found at all surprising, but then there was extreme skepticism that what we found was of any significance whatever. And eventually we published a paper in the Ember Journal. Because of all this skepticism, I didn't want to get things wrong. There was a big tradition of not getting things wrong. But anyway, I decided to publish the paper. And actually the person who submitted it on my behalf said it was very interesting, but of course wrong. But one of the editors thought not, actually, and it's the only time an editor has rung me up to discuss a paper that I'd submitted, and said he was extremely interested in what I'd done. He thought it was highly significant. So there it lay for years, and I used to think, oh, you know, one day this thing's going to come out, and it did, and it turned out that we were correct, and it had highly predictive properties, and if one finds these motifs, one can be pretty sure that they've got something to do with binding a purine nucleotide, which is very important in biochemistry. So I mean, I didn't call them. There were two motifs, A and B, and people now call them Walker A and Walker B motifs. So I've been
fame, immortality.
And actually somebody came up to me at a recent conference and said, you're not the Walker of the Walker motif, are you? And I said, yes. They said, oh, you're still alive.
Yes. I thought you were
so
dead.
I thought full circle, you know.
That's right. Well, another chemist who I interviewed, who had, I think he had a metal or ore named after him, a murigite. This is John.
Oh, this is John Myrick
Thomas.
Yes, right.
Do you know John?
Well, I met him particularly in recent years through the chemistry department and through his association with a biochemist friend of mine, Leslie Dutton, who was one of his PhD students. And then because of John's friendship and knowledge about Max Perutz, and so we certainly talked about Max together, and I remember some of the tributes that John made to Max around the time of his Max's death. So I have met him from time to time.
Very loquacious.
Very loquacious, yes.
With a lovely bass voice.
described Max as being pellucid. His writing has been pellucid.
Yeah. But John talked, and this is something I would perhaps like to now, unless there's anything else we should say about the Nobel Prize or your work.
Whatever you want.
Well, John talked quite a bit about Cambridge as a place to do research.
Oh yes.
You mentioned the community, which gave you support. A number of scientists have said that they've been tempted to go to America, and of course Sydney did, and Francis did, to a certain extent. Have you ever been tempted to go away?
Not really. I mean, the environment at the laboratory of molecular biology was, and still is so stimulating. And of course, one of the great attractions was having long-term support that one justified via quinquennial review every 5 years, and so effectively one didn't need to spend a lot of time writing grants, so that's hugely attractive to me. And although I enjoy the little teaching that I've done, of course it meant that one was free from any teaching responsibilities as well, and so we are full-time researchers in the Medical Research Council, so we are different from a university department in that respect.
When you say the little teaching, do you lecture? Have you lectured at all?
I have. I was always impressed that Sydney would give a course of lectures to undergraduates, and that Max did as well. And I went to Max's lectures early on, and so I decided that I'd like to give some on bioenergetics, and so I used to in Part 2 biochemistry. It was part of the core curriculum. We now evolved into an option, so we still do it actually. We give a 12. I just used to give, I think, 2 or 3 lectures myself, but now it's evolved into a course that involves several of my colleagues at the DUN. It's a 12-lecture course. I give a couple of lectures, the introductory lecture. And I find that a good thing to do from many points of view. It gives contact with the students, they know we're there out of the Adbrook site, and I enjoy that contact.
What about PhD students? Have you had?
I've had a lot of PhD students over the years, and more students than postdocs, I think. And at the DUN we have 50 PhD students, and I take a lot of effort over them. I think it's very important for the success of the unit, because a lot of the research is done by students, and we're dependent upon them. So I enjoy that interaction very, very much.
And then you, I don't know if you had a college attachment before you went to Sydney.
I didn't, but I had, strangely enough, taught at Sydney Sussex College, because a friend of mine in the biochemistry department, Peter Henderson, was a fellow of Sydney, and he went on sabbatical and asked me if I'd like to take over his teaching. And I think this would be the early 1990s, or perhaps late 1980s, and I thought about it, and I decided I would, because I thought it would be a very good opportunity for me to, as it were, relearn the subject, because I was conscious that having worked for 15 years or so in the research institute, I, as it were, knew more and more about less and less, and so I thought that I'd like to relearn the subject, and I found that extremely valuable. I think I did that for a couple of years. I taught first year students in the cells course in Sydney, thanks to Peter. That's the only time that I've done that kind of teaching.
Then you became, Gabriel Horn, I think, invited you to be a fellow of Sydney.
He did, yes.
In about 97, was it? Or?
It was post-Nobel Prize, immediately, pretty soon after Sydney. Gabriel invited me to become a fellow, and I became a fellow, I think, before I received the prize, so it was between the announcement and actually the award.
Did you enjoy Sydney?
Very, very much, actually. I knew that it was something that was missing from my life, having been an undergraduate and graduate student at Oxford, and having had a lot of friends in arts subjects. I was quite conscious that at the Laboratory of Molecular Biology, I was spending most of my time with other scientists and having very little contact with people in other disciplines. So it was something that I welcomed, actually, and it's the aspect, one of the aspects that I enjoy most about being a fellow at a college is meeting people from other disciplines and talking to them about what they do, and for them wanting to know what I do. And I found the collegiate nature of Sydney, Sussex extremely pleasant, actually.
What about your home? I mean, did your wife also go to Sydney sometimes? I mean, I think they'd probably be, because when Prill was there...
Yes, well, they were very good at looking after fellows' wives, and I think it was probably Gabriel who instigated an annual dinner with the master and wives and partners, which my wife joins in college activities as she's able to, and also enjoys it.
There are three more general questions I had. One was, you mentioned earlier on your enthusiasm for music. Does that still mean a lot to you, music?
It does, yes. I mean, I still enjoy singing, although I've but it's in recent years I've really not taken part in any singing activities other than the occasions when I go to church. But I always enjoy singing. I enjoy going to musical concerts. I listen to music constantly, more or less, when I'm not working.
What kind of music do you like?
I've got a fairly wide taste, but you know, the usual suspects. I guess I was sitting listening to some Beethoven, Schubert. I was listening to some Bach. I enjoy that. I enjoy opera. So I've had a lot of associations over the years with Italian universities, and I decided it was actually George Rada, who was the chief executive of the Medical Research Council, put me up to this. He said, you know, when I go and give lectures in Italy and in Europe, I don't ask for a fee, but I insist that they take me to the opera. So I got onto that idea immediately, and so I love the opera, particularly Italian opera.
Rada is spelled R-A...
R-A-D-D-A.
Right.
Yeah. But I also enjoy other forms of music. I as an undergraduate, I was interested in folk music and did Morris dancing and folk singing. Became squire of Oxford University, Morris dancers and all this sort of stuff. And then I enjoy composers who then are like Vaughan Williams and Holst who've used folk music themes in their works. So and then my wife has always been very keen on traditional jazz, and that certainly brushed off on me as well. So fairly wide taste.
The second question, you mentioned the Vietnam War and some ancestor who was conservative. Have you been politically involved much, or have any strong political associations?
I belonged to the Labour Club when I was an undergraduate. No, I can't say that I've had strong political formal associations. I've always felt that I was, as it were, too busy to get involved, or too committed to other things to get too involved in that. I mean, I think if I have sympathies, they would lie closer to the left than to the right. Well, no question about that. So, you know, pretty much in favour of broadening access to Oxford and Cambridge to state education. It's an issue that concerns me very much, in the sense that I've often wondered whether somebody from my background would find it more or less easy to make it to Oxford and Cambridge than I did in 1960, and I'm not sure that I know the answer to that. So I mean, issues like that do concern me.
And we also, you mentioned an early association with chapels and the Church of England.
Yes, and well, I mean, like most people who become interested in science, I think inevitably there becomes a conflict in your mind between the religion or the Christianity that you've been brought up with and part of you, and the logic of science, and that that has to be resolved. And so that took me slightly longer than some people to resolve it, but I came under fire. I tried to protect my position as a believing Christian as an undergraduate, came under fire from people who really made me think about it, from the students that I knew, and eventually I decided that they really were incompatible, and so I, as it were, lost my religion, and that's still my current position. But having said that, actually something I have discussed with Gabriel, I still enjoy going to chapel and taking part in the service. Of course, one isn't participating properly in what is going on, but it's rather like participating in a kind of ritual, which I feel is part of me actually, and I can't resolve. And actually, I've decided I don't want to resolve it. So, I've never met Richard Dawkins, but he would. I think he'd give me pretty short shrift for retaining those views. But I do feel it's just part of my makeup, and I don't want to get rid of it. I get some kind of enjoyment. Enjoyment is not the right kind of word. It's not some kind of vicarious enjoyment. It's actually it is spiritual in a way that I can't fully explain. And so I make no apologies at all for from time to time going to chapel in Sydney. And we actually have very fine music now in Sydney. We appointed a new music director last year who was extremely successful and has raised the standard of music to a very, very high level.
What about reading? Do you read poetry or novels or?
I read novels. I read quite a lot of biography actually. I'm sort of perhaps it's part of getting older. I'm interested in how discoveries were made and who did what. So I read recently a biography of Fred Hoyle that I happened to spot, which turned out to be really interesting. I read another one, biography of Lord Kelvin, recently. So I'm interested in the history of science, and I'm interested in history in general. I feel I don't know as much as I would like to about when things happened, and I've been reading about the Stuarts recently because that wasn't part of the history that I learned in school. My history started in 1714 and went till 1918, so I know quite a bit of that, but I never quite understood the Tudors and the Stuarts, so I've been reading some of that recently. I'm still very much imbued with this idea that comes from my mother that you should be improving yourself all the time. Of course, all academics feel that way, that we want to go on broadening ourselves and learning about things, but I do feel that quite strongly, that I don't want to waste my time. I want to learn about things that I didn't know about. I'm trying to learn Italian at the moment to improve my ability to listen to Italian opera, and so I think I do read quite widely.
Well, all these interests, you've got many interests. We're at the same age, a few months apart, and I'm retiring in 2009. I'm not sure whether you reach 67.
I have reached 67 just last week. I have a contract that runs till I'm 68, and just what will happen after that isn't clear. I'm in discussions with various bodies, but my intention is not to retire. As long as I'm physically able, I would like to continue certainly longer in something which is close to laboratory science. I think that's what what I do best. Over the past 10 years I've had to assume an administrative role, and I have enjoyed building up the done and getting people there and bringing along younger people. But I'm quite interested in continuing my own research. I've managed to keep it going during this period, and I would like to continue doing that. I think I'm still reasonably productive, and I feel there's a bit more in me yet, and I'm not ready to go out to grass yet.
As the joke says, retirement is for younger people. A question I often ask at the end of these interviews is, if you were giving advice to a young scientist who was just starting out, I asked Martin Rees this, I think, is there any particular thing you would stress to them? I mean, many have said that it's very difficult times to start out in science now, but
it is, because so many other careers appear attractive, and it's another thing that worries me is that many of the best minds are just not attracted by science anymore for financial reasons. They see themselves having a better life by doing something else. But for somebody who is committed, I think if you're going to be successful, at least in research in science, it's very, very important to find the right topic to work on. And now, exactly how you do that, I think, is difficult, and it's much more difficult than it was 20 years ago, because fields of science are the ones that I work in are much more heavily occupied, and so finding your niche is actually quite difficult. So I think thinking carefully about the area in which you work, and then committing yourself to that area is very important. I think I would undoubtedly want to work in some aspect of neurobiology today. There are so many unknowns there that I think one really could make make a niche, an important niche, and make real contributions in that area. So I think that's one piece of advice I give.
Not to go west, young man, but go to neurobiology, young man or lady. Anyway, thank you very much indeed, John.
That's my pleasure. What did Martin say?
I'll have to look at the interview. It was something about, it was again something probably to do with finding good problems to study, and commitment, and I'm going to collect together all the different
bits of advice.
And
I've not been asked that one before. I mean, one gets used to answering the same questions. What impacts the Nobel Prize, how would you like, all this sort of stuff. So one has that a little bit off pat, but that one I've not been asked before.
Sense of humour sounds as if it might be useful as well.
I think so, yes.
Anyway, thank you very much indeed, John.